Warm greetings, my dear students. Welcome to the Physics Clinic at the Red Rose Academy. In continuation with the last class, we are seeing the packaging, right? How the packaging takes place, right? So we'll see all these things right now. Yesterday, I suggested so many questions to you, right? I taught you so many questions like how many, I gave you an assignment like how many nucleosomes, okay, how many nucleosomes are present in mammalian cell, like that I gave you. So right now you should understand, okay, you know how to calculate, okay, it is although not possible to determine the number of nucleosome present in the mammalian cells. But in case of human, approximately 30 million nucleosomes. 30 million nucleosomes present. Okay. So you must understand that 30 million nucleosomes are present. Okay. Then how the packaging takes place in prokaryotes and eukaryotes. You know about prokaryotes. Prokaryotes nucleus is absent, right up. And like E. coli, we can say that is prokaryote. So prokaryote. Okay. So it is no nucleus. Okay, nucleus is absent. Got it. So, but the DNA is expressed in the form of large loops, okay, held with some positively charged proteins to form nucleides. Okay. So it is basically negatively charged. The DNA is negatively charged here. The greatest example is E. coli, Escherichia coli. And what about eukaryotes? You know, eukaryotes consist of nucleus. Okay, nucleus is present, and you know, it also consists of DNA. Okay, then that DNA it tends to. Okay, so I will explain you. Okay, in eukaryotes, how the packaging takes place. Okay, we know that in in prokaryotes, the DNA is present in the form of a large loop kind of thing, held with some positively charged proteins. Okay, since that is negatively charged, DNA is negatively charged, it is present in the form of large loops held with some positively charged proteins, okay, to form nucleoids. So this is actually what? Prokaryotes. What is an eukaryote? Eukaryote, you can say how the packaging is. Just take down histone octamer plus DNA. It gives nucleosomes, okay, chromatin, okay, chromatin fiber, and chromosome. Okay, so what is this histone octamer? It is made up of 8 molecules of histone proteins. Okay, histone proteins are positively charged basic proteins. That is histone proteins. These proteins are rich in basic amino acids like lysines and arginines. Okay, both of these amino acids possess positive charges in their side chains. Okay, what are the basic amino acids? We can say lysines and arginines. Okay. So then I told you this is histone octamer. Then nucleosomes. How the nucleosomes will be? You know, DNA is the negatively charged one. It wraps the positively charged histone octamer. Okay. It is actually shown in the figure. I'll show you that figure actually from your book. One second. See this one. This is what I want to explain. Okay, this is very, very, very important. You must understand how it is formed. Okay, this structure. Okay. In a nucleosome, you can say how it is formed. Okay. So I told you DNA wraps the positive charge. This is DNA. Negative charge. This is negatively charged. Okay. So DNA is negatively charged. It is wrapping the histone octamer. Okay, this green color is histone octamer. Can you able to understand? As shown in this particular figure, we can understand how it is formed. Okay, then chromatin. I told you, right? Chromatin. It is a thread-like colorful structure. It is present in which repeating one or more nucleosomes with DNA in between. Okay, so under electron microscope, chromatin appears as like this, beads on string. It appears like this. It is chromatin. Okay, and it is the electron microscope picture. This one is the electron microscope picture. Here you can see, okay, beads like structure. Okay, so it's really the nucleosome. Okay, then chromatin fiber. So further packaging of this, it results in the formation of chromatin fiber. These fibers are further condensed and developed during metaphase to take the shape of the especially the shape will be chromosomal shape. Okay, for this packaging, non-histone chromosomal things are required. Okay, non-histone. Okay, for packaging the chromatin fiber. Okay, then we'll see euchromatin and heterochromatin. You know what is euchromatin? So arrangement of chromatin material in a nucleus. Okay, this is what this concept of euchromatin and heterochromatin. Okay, what is euchromatin and heterochromatin? This is I will explain you. Okay, see, euchromatin and heterochromatin concept is how the chromatin material in a nucleus is arranged. Okay, so it is arranged in two ways. One is loosely packed chromatin or euchromatin. Euchromatin is loosely packed chromatin. Okay, wow. And it is transcriptionally active. Okay, it is very, very active. Okay, it stains light. For example, we can say the chromatin that is more densely packed, that is heterochromatin. Okay, it is said to be transcriptionally inactive. Okay, so this two difference you have to understand what it is. Okay, euchromatin is transcriptionally active. This heterochromatin is transcriptionally inactive. This and all very important. Then in your book it is given the search for genetic material. Okay, so Measure, he only proposed for the principles of inheritance so many things. Okay, in 1926. Okay, to determine the mechanism for genetic inheritance. Okay, it reached the molecular level. Okay, previous discoveries by Mendel, Walter, Thomas, other scientists had narrowed the search to the chromosomes. Okay, right now, this particular thing. Okay, we can say what molecule or actually what material was actually the genetic material. Okay, had not been discovered. Okay, the question is like that. Then I'll say by Frederick Griffith in 1928, transforming principle by means of this. What is the transforming principle? It is a series of experiments with bacteria. Okay. So this bacterium responsible for pneumonia, it says, right? Streptococcus pneumonia. In 1920, this particular person, Griffith, he has done so many series of experiments on the bacteria named, what is this? Streptococcus pneumonia. Okay. And during this course, Griffith has observed the change in the physical form of the bacteria during this experiments. Okay. So it is given like that picture. It's not given here. So it's a search for genetic material. So it's a very great search. Okay. I'll show you some pictures so that you can able to understand the transforming principle. I think that picture also in your book. Let me explain you about this experiment. Okay. And this picture clearly shows, okay, what he has observed. The bacteria causes pneumonia. Okay. So what is the main particular bacteria? So we studied about Streptococcus pneumonia. It produces two types of colonies. One is smooth, okay, smooth and shiny colonies. That is S-strain or virulent. Okay, S-strain or virulent strain. S-strain or virulent strain. Okay, then this bacteria possess mucus. Okay, polysaccharide coats. Then rough colonies, that is R strain. R strain is rough colonies. See, smooth and shiny it will be. This is rough colonies. Bacteria lacks mucus coat. Okay, then so S strain now injected into the mice. Okay, then mice, what will happen? This is S strain. Okay, you can able to see. S strain is now injected into the mice. Then mice die. Then R strain, it is injected into the mice. So mice lives. Okay. Then heat killed estrine. So it is heat killed estrine injected into the mice. So mice lives. Then heat killed estrine plus R strain. Okay. So heat killed estrine plus live R strain. Okay, both injected, then mice dies. Okay, so he observed all these four steps. Okay, I'll explain you, I'll repeat this again. These are all the bacteria, keep in your mind. Okay, so for rough strain, they have quoted like this. Okay, rough strain, live. Live means, it is the live bacteria. It is the, alive bacteria. Live bacteria or strain is injected into the mouse live. And estrine is injected into the mouse dies. Okay, then heat killed estrine is injected into the mouse lives. Then both heat killed estrine, okay, and live or strain injected into the mice or mouse, the mouse dies. Okay, so this is the experiment, greatest experiment he observed. Okay, so Griffith, what all these four steps? Okay, in the step 4, he recovered live estrine bacteria at last. That's what happened. So this clearly indicates that the live or strain has been transformed into the live estrine bacteria. That's what he observed from this experiment. Okay. So this transformation becomes possible due to the transfer of some transforming principle. Okay. That is, should be genetic material. Can you able to understand? This is the very great history he found. Okay. So therefore, how actually killed a strain bacteria and live or strain bacteria is injected to the mouse. Okay. Then mouse dies. Okay. Then finally, in the fourth step, Griffith recovered the S strain bacteria. Okay, at last, this clearly shows that it is possible to transform, okay, from uh, live R strain to live S strain. So there is the transforming principle here. So this is said to be the genetic material. How it is live? So R strain becomes live S strain bacteria. So there is a transforming principle takes place, genetic material. So that is the genetic material. Because of this transforming principle, R strain was able to develop a smooth polysaccharide cover and become virulent. Okay. By this experiment, it was proved that some genetic material has been transformed from heat killed A strain to R strain, live R strain. Okay. But the nature of the genetic material was not established here. Okay. Then characterization. What are all the characterization given in your book? Let us see that. What are all the characterization? See, biochemical characterization of transforming principle, it is given here. So many genetic materials. Actually, previously it was thought to be protein. Okay, this Oswald Avery, Colin MacLeod, and Macklin McCree so worked out the biochemical nature of the transforming principle in Griffith's experiment. Okay, so what was the result we are going to see? They extracted and purified the biochemicals, DNA, RNA, proteins, etc., obtained from the S strain of bacteria. Okay. Therefore, these biochemicals were used separately to prove which was acting as transforming principle or genetic material, we can say. Very great idea, right? Students, you must understand they extracted and purified the biochemicals, that is DNA, RNA, proteins, etc., obtained from the heat-killed S strain of a bacteria. Therefore, thereafter, these biochemicals were used separately to prove which was acting as transforming principle or genetic material. Okay, what is the step one we saw? DNA extracted from heat-killed S strain plus R strain injected into the mice. Okay, what is the step one? Heat killed. Uh, okay, so can you write this? Then it is easy for you to understand. You can write as a tabular column. What is the step one? You can write steps. Okay, step one. Step one is DNA extracted from heat killed or strain, sorry, S strain plus live or strain. Okay, injected into the mice. Then what will happen? Mice dies. Okay, mice or mouse, anything. So mice die. Then DNA is the genetic material. So these are all the process. Okay, DNA is the genetic material. Okay, so this is the first step. What is the second step? Like this, you can put 4 column. Okay, and then write all these things. Okay, then the first step is over. What is the second step? Second step, can you write now? RNA, these are all the genetic materials we need to prove. So RNA extracted from the same heat killed S strain plus R strain injected into the mice, then mice lives, mice living. So RNA is not the genetic material. Then step 3 is protein extracted from heat killed S strain plus live R strain injected into the mice live. So protein is not the genetic material. Then step 4, you can say heat killed S strain plus R strain plus we can say DNases, all DNases injected into the mice. Then mice live. Mice is living. Okay. Then DNA is the genetic material because along with that, DNase is also injected into the mice. So that mice is living. Okay, so DNA is the genetic material. So that is how it is confirmed. It is proved. Okay, so what is the step 5? You can write now step 5. In step 5, heat killed S strain plus R strain plus all RNAs is injected into the mice. Then mice die. So, or confirm it is RNA is not the genetic material. Then step 6 is heat killed S strain plus R strain. All proteins, okay, proteases like that, proteins injected into the mice. So mice die. Protein is not the genetic material. So plus and minus, we can say all these things. Okay, so confirmed DNA is the genetic material. So by the above experiments, all these guys, that MacLeod, McCarty, and Avery, so they concluded DNA is the genetic material, but many biologists disagreed with them. Okay, so we'll see the next topic. So right now it is confirmed DNA is the genetic material. You'll be asked, okay, I told DNA is right. So what is DNA? What is DNA is? DNA and DNAs. You must understand all these terms. DNA is the nucleic acid, which is the hereditary material in all organism except few viruses we saw. DNA is deoxyribonuclease, which is a protein. Okay, that one DNA is the nucleic acid, deoxyribose nucleic acid, which is the hereditary material. But this is deoxyribose nuclease, which is a protein nuclease. Okay, catalyzes that hydrolytic cleavage of, you know, all these things, right? What is that? Hydrolytic cleavage means that hydroxyl group is now broken. I mean, it is cleavaged, it is broken. Okay, of phosphodiester linkages in the backbone of DNA. That protein, this protein only catalyzes that hydrolytic cleavage of phosphodiester linkages in the backbone of DNA. Children, all you have to you have to take a note. This is very important and keep in your mind. Okay. So right now we saw that DNA is the genetic material. So bacteriophage, we can say. Okay. This was the first experiment by which all biologists were convinced that DNA is the genetic material. Okay. What was that? In 1952 also it was carried out. Okay. Alfred Hershey and Martha Chase. They worked with viruses that infect bacteria called bacteriophages. Okay, this is very important experiment. Next to the previous experiment we saw, this is the bacteriophages. This experiment is done with. Okay, and it was the first experiment by which all biologists were convinced. Okay, in the first experiment they were not agreed, but in the second one they were agreed. Okay, so a bacteriophage is a virus that infects bacteria. That is bacteriophage. Okay, keep in your mind. What is the life cycle of bacteriophage? Bacteriophage is virus attaches itself to the bacteria. The genetic material of virus we need to see. Okay, the genetic material of the bacteria, it is entering into the genetic material of the virus, it is entering into the bacteria. Okay, so virus genetic material into the bacterial DNA to synthesize more copies of virus. So a lot of viruses come out after this kind of bacteria. Okay, so then I'll explain you this. See in this is the bacteriophage. This picture clearly shows, okay, all these things. Hershey Chase experiment, we can say. Okay. So what are all the steps involved in this Hershey's Chase experiment? I will explain you right now. This is Hershey's Chase experiment. Can you write all of you? Then only it is easy for you to understand. Okay, what are all the steps involved in this Hershey's Chase experiment so that you can able to understand what it is. Okay, you can put the table like here it is steps. Okay, then part of the virus becoming radioactive. part of the virus becoming radioactive. Okay. Then you can say attachment, like infection, first attached and then infect and then blending, then centrifugation. Centrifugation means it is like a mixer grinder, it shows, right? It is just centrifugate. That is meant by centrifugation. That grinding, like, okay. So after centrifugation, radioactivity. After centrifugation, these are all the, we can test this in a blood only, right? So samples, okay. Radioactivity was observed in, okay, it was observed. And we can say, what is the inference? That also we can understand. Okay, what is the whether DNA is the genetic material or not? Okay, so like inference we can say. In all research we can say like that. So what is the first step? Can you write now? The first step: virus grown in medium with radioactive phosphorus. That is P32. Okay, virus grows in medium with radioactive phosphorus. That is P32. In the part of the virus, you can write DNA becomes radioactive as it possesses sulfur, and the protein will not become radioactive as it lacks sulfur. So, what you have to write in this part? DNA becomes radioactive as it possesses sulfur, but the protein will not become radioactive as it lacks sulfur. Okay. Then how it is attached, infecting, blending, configuration, centrifugation. Centrifugation means separates the virus coats from bacteria. By the mixer grinder, it moves like that, right? So what is that processor, food processor and all, like that it will be done, and then the separation between the virus and the bacteria. Okay, and after settling, two compartments were formed: upper of viral coats without genetic material and lower bacterial with genetic material. So keep this in your mind and take a note. Actually, in this centrifugation, it separates the virus coats from bacteria, and after settling, two compartments were formed: upper of viral coats without genetic material and lower bacterial with genetic material. Clear? Then after centrifugation, what will happen? Radioactivity was observed. So in that column, you can write the lower bacterial compartment with genetic material indicates that viral DNA has entered bacteria. That is the indication. So the lower bacterial compartment with genetic material indicates that viral DNA has, entering the bacteria. So what is the inference we can say? So DNA is the genetic material. From this we can confirm. The second step you can write now: virus grown in medium with radioactive sulfur. First radioactive phosphorus, next radioactive sulfur, S35. Okay. Then in this, okay, what is the part? Which part is becoming radioactive? Protein will become radioactive. Take down, all of you, as it possesses phosphorus, but DNA will not become radioactive as it lacks phosphorus. Okay. Then centrifugation normally like this. Then here, after centrifugation, what happened? The upper compartment of viral coats without genetic material indicates that viral protein has not even entered the bacteria. So this is the only proof protein is not the genetic material. Okay. So this is also confirmed. DNA is the genetic material. Protein is not the genetic material. But previously it was misunderstood like protein was the genetic material. But by means of all these experiments, the scientists proved that DNA is the genetic material. It was done in 1952 only. Okay, it was carried by Alfred Hershey and Martha Chase. So this famous experiment is known as Hershey-Chase experiment. Okay, it's a very, very important experiment. You must know. Okay. Then we'll see. If you want to write DNA versus RNA, then there are so many properties you must know. Okay, so you can write DNA versus RNA. You must compare. So everything given in your book very in detail. Okay, you can go through it and write all the differences. Okay, and I want to confirm with you all the properties. Okay, genetic material, if you take, it should be able to replicate. That is the first criteria. Okay, if a molecule to act as genetic material, I will say this to you. Okay, criteria. Can you write now? Criteria. Okay, criteria for a molecule to act as a genetic material. Molecule to act as a genetic material. Is it clear for you? What is the first point? First point is it should be able to replicate. It should be able to replicate. Replication is very important. Replicate. It should be able to replicate. Second one, it should be chemically and structurally stable. Okay. So what is the second point? It should be chemically and structurally stable so that it may resist any change due to changing environment or age or physiology of an organism. Okay. So these two criteria you must understand. And also it should slowly undergo mutations. Okay. You know that, right? Mutations. It should slowly undergoes. And also it should be the important source of evolution also. Okay. And the fourth criteria I can able to say, it should be able to express itself in the form of Mendelian characters. Itself it should express. Okay, Mendelian characters. You know all the Mendelian characters. Okay, dwarf, recessive, and tall, dominant, everything. So this has to be maintained. Okay. So these are all the criterias. I told you about the four criterias. I hope you wrote all the criterias right now. And also you have to learn how the DNA and RNA is differentiated. So comparison of DNA and RNA as genetic materials, how it will be. So that is very important for you. Okay, DNA is, if you want, I will explain you this also. You must know both. Okay. Let me dictate, you can write. Okay. So replication, both replicate. DNA and RNA. Like this you can put. Here you can write the features. Okay. Like this. Features, DNA and RNA. First point, you can write replication. Self-replication, right? Self-replication. So both will replicate. Then stability. If you look at stability, that 4 points I discussed, right? So based on that. Stability, you can write DNA is chemically less stable